The Amazon rainforest holds great importance to the world's biodiversity and environmental heritage. The Amazon, also known as the Earth's lungs, is home to the most rich and unique plant and animal species in the world. However, this biodiversity is currently under threat, but not only from deforestation. A silent threat lurks beneath the canopy, and this is selective logging. Selective logging is a process of removing one or two particular species, in this case the timber species Dipteryx odorata and Jacaranda copea, while leaving other surrounding species untouched. So selective logging is far more difficult to patrol or monitor than large-scale deforestation, and can often lead to serious implications in the loss of genetic diversity, reproductive capacity, and ecological functions.
The two main concepts that Vincent and his colleagues explore in this experiment include inbreeding and gene flow in relation to two tree species, Dipteryx odorata and Jacaranda copea. Inbreeding is the mating of organisms that are closely related that result in offspring. It is commonly seen in plants via self-pollination and promotes homozygosity in the population, i.e., the alleles are the same at a location. Inbreeding can be useful in ways such as keeping desirable characteristics in a population and eliminating the undesirable ones. However, one of the major downsides of inbreeding includes a decrease in biological fitness. This can be through a decrease in the size, vigor, and fertility of the offspring, and this is called the inbreeding depression. Therefore, a small gene pool will negatively affect the biological fitness of a population, while a larger gene pool is a lot more diverse in its genes, and therefore is a lot more robust. Inbreeding can also lead to the expression of deleterious recessive alleles. These are deleterious mutations that occur on two variants of the same gene at the same location on a chromosome, i.e., an allele. These are recessive and are hidden in heterozygous individuals. These are individuals that have two different alleles at the same location. However, these deleterious alleles are expressed in the homozygous individuals. Genetic flow, also known as gene migration, meanwhile, is the introduction of new genes of or alleles from one population to another. The introduction of these new genes or alleles into a population not only increases the gene pool, but also increases the variability. Therefore, new combinations of traits can be possible. This migration of genes in and out of a population results in the changes to the allele frequency. This is the amount of members carrying a particular variant of a gene in a population. There are a number of factors that can increase the genetic flow, with the main one being mobility, and in this case, how mobile the seeds are in these two tree species. However, there are also a number of different barriers to gene flow, with the main one being physical barriers. These can either be naturally occurring ones, such as oceans or mountain ranges, or can be man-made, such as the Great Wall of China. Other barriers include incompatibility and non-viable hybrid offspring.
This study aimed to look at the effects of selective logging on two Amazonian timber species in the same forest, Dipteryx odorata and Jacaranda copea, with contrasting ecological and reproductive characteristics. This study specifically looked at the impacts of logging on genetic diversity, pollen flow in both high and low density timber species, and on gene flow in synchronous and asynchronous flower species. Genetic diversity and gene flow were characterized by genotyping adults and seed samples both before and after logging, using hypervariable microsatellite markers. The study sites differed in size, with the study site for Dipteryx odorata being 546 hectares, while the site for Jacaranda copea was 200 hectares. The selective logging for these sites was carried out from September to December in 2003, in accordance with the legislation at the time. Overall, close to 50% of the trees of each species were removed. The tissue samples were collected from cambium, fruits, embryos, and seedlings, and the DNA was extracted. DNA was genotyped using polymorphic microsatellite loci, which is a repeated small section of DNA, commonly used for paternity tests. Seeds were sampled from the same mother and from different mothers to compare the difference between these groups, as well as to test the difference between years. A number of analyses were carried out for both of the species, including a neighborhood analysis, a paternity analysis, as well as genetic analyses, including array analysis on the seeds. The genetic analysis was performed before and after logging. Also, the assessment of short-term impacts of selective logging on the pollen flow was carried out. This study found that in Jacaranda there were no short-term genetic impacts on the population, with very little allele loss. It was found that the species had high outcrossing rates, both pre- and post-logging. Within Dipteryx odorata, it was found that selective logging showed a range of genetic impacts. These impacts included a 10% loss of alleles and reductions in breeding via pollen from trees within the study area, as well as in the number of pollen donors per progeny array. For both of the species, they found an insignificant difference in the allele frequencies between pre- and post-logging samples. The pollen was found to have dispersed over large distances, and that the majority of pollen was found to have come from outside the study area. While both species did have losses of alleles, it was also found new alleles had immigrated from outside the study site.
Selective logging has seen long-term ecological changes, including structural transformation of our forests. This has seen incidences such as an increase in reed growth due to the thinned canopy allowing for increased light interception. As a flow-on from this, there has been a reduction in the diversity of local plant species. Alongside this, carbon sequestration is a major issue being tackled today. However, logging counteracts these efforts as carbon storage is reduced with the removal of trees. This transformation could pose as a threat to forest health and surrounding agroforestry systems, and thus the way in which selective logging is implemented must be reviewed. To reduce the risk of degradation and forest structural transformation, improved logging programs should be considered. These programs will aim to reduce inbreeding and promote gene flow throughout the logged timber species. To achieve this, these programs will include selective logging intensity thresholds in conjunction with further research into the true implications of this practice. Academics from the University of Adelaide suggest that unlike deforestation, little is known about the greater impact selective logging has on biodiversity, reproductive capacity, genetic diversity, and ecological services. It is believed that selective logging may be a large contributor to the disappearance of the world's tropical forests. This is because this practice is often found where deforestation is a threat. Thus, sustainable logging practices can be implemented and diffused to locals with newfound forest-specific knowledge, thresholds, and policies in place to protect these highly valuable ecosystems. The Amazon rainforest is suffering at the hands of human malpractice. While selective logging is not currently perceived as a pressing issue, it has the potential to induce irreversible change to the world's tropical forests. Already we have seen a slight reduction in genetic diversity with the minor loss of alleles in selected timber species. While this is no significant loss yet, in the future it could be a greater concern in reproductive capacity and inbreeding. Because of this, there is a push for the development of more sustainable logging practices through thresholds, policy, and future research. Action must be taken to prevent selective logging becoming a major degrader of our Earth's lungs, because if we don't, we may suffer one of the world's greatest environmental disasters yet.